So yeah, that should be better. Yeah. So good evening. Right now it's evening for me. I hope you're having a good day today. So for today I'm presenting my work, Genomic Characterization of Bartonella species associated with Carrion disease, and insight into the evolution and virulence of this species. My name is Camila Castillo-Vilcahuaman, and I'll be presenting my work for today. So first, like a brief introduction of what is Bartonella bacilliformis, and more importantly, what's Carrion disease. So Carrion disease is it's a very restrictive and neglected disease that is exclusively located in the Andean region. So it's basically a disease that will only present on Peru, Ecuador, and probably Colombia. Um, it has been documented for a long time in the history of these countries, and you can even see archaeological remains that depict how this illness affected the native population here. Carrion disease is mainly produced by this pathogen here. This is an electrophotography of an electron microscopic photography of Bartonella bacilliformis. As we can see, it's actually a flagellated bacteria. It mostly possesses, I think, like 6 or 12 polar flagella. It's a very small prokaryote. It's intracellular and it has a short genome based on the lifestyle it has. Carrion disease is characterized because it has two phases. So first you have a skin disease that's presented like eruptions on your skin, and you can also present a high fever. For a lot of years, people believed that these two were two separate illnesses. However, Daniel Carrion, the man that gives this disease its name, discovered that these two diseases were actually one, by doing a very, let's say, heroic thing that is still remembered in its country today. Um, so how does Carrion disease work? It's mainly transmitted by a blood-sucking arthropod that will pinch at a human skin. This disease, by the way, it's mainly hosted by humans. And what the arthropod will do is to pinch and lay the bacteria, infect you with the bacteria, and this bacteria, what will do is try to invade their erythrocytes. This is mainly a bacteria that lives in that invades erythrocytes, and its main work of action starts from here. Um, so the whole Bartonella genus, that's what we've seen here, is mainly characterized by three very distinct groups. The first one is here, Bartonella apis. It's the only non-pathogen in this whole genus. It's a symbiote. I remember it's a symbiote from bees, and it lives as a symbiote in bees. It's not a pathogen, and it's mainly restricted to that lifestyle, let's say. The first surgence of pathogenicity comes for here in Bartonella tamiae. This is the first appearance of pathogenicity in the history of the evolution of the Bartonella genus, and what we can see here is that Bartonella tamiae will have a very distinct, let's say, way of developing a pathogenicity, because after, or let's say that from the ancestor that Bartonella tamiae had in common with the other Bartonellas, there's another resurgence of pathogenicity that it's mainly represented in this diversification here from the Eubartonella group that comprises all Bartonella species that are not Bartonella termii nor Bartonella apis. And the Eubartonella group is divided in 4 groups, being the lineage 1, lineage 2, lineage 3, and lineage 4, the groups that comprise the Eubartonella group. Our subject of today is mainly in this lineage. It is only in this lineage. It's the Bartonella bacilliformis and its close relative Bartonella cashensis, although they are very separate from each other, and we will see why in the following presentation. If we search for Bartonella bacilliformis as a species in the NCBI database, this is what we'll get. These are all the strains that are currently present on the NCBI, and something interesting here is the fact that we've got here 2 branches, right? This is the branch from where all of the other strains diversify, and here we find the per 097 strain. This one is the main character, let's say, of the whole study we are presenting today. We can see why. It's kind of an outlier in a very close group, really. It's when you compare all these strains, they are very similar to each other since, well, they are intracellular and there's not really a whole lot of diversification going on, or that's what mostly people will believe. However, we have here an outlier that's kind of presenting itself as something a bit different compared to the others. A lot of work has been done by doing molecular characterizations of these species. And what's interesting for us is the fact that Bear 097 has been time and time again mentioned as a very curious strain that nobody knew really what was going on with it. So we've got it as, for example, a subspecies, like in this introduction here. However, this same paper mentions that other papers believe that this might be another species completely. Probably one of the reasons why we couldn't really get into how this or what's actually classification going on here is mainly because of this, the fact that from all the strains that are available in the NCBI, the Bear 097 is the only one there. There are no more Bear 097 strains here. So probably this led to the fact that people can only speculate based on one genome. So for us, our main objective is to try to elucidate the taxonomic status of Bartonella bacilliformis by trying to exclusively examine its genomic composition and do several comparisons in this context, in the genomic context. We hypothesize that the variations we see in Bartonella bacilliformis are actually reflective or reflect the significant divergence that exists in this group, and it might indicate the presence of 2 species rather than just one. I know this might be a little bit very out there, but at least we try to remain in the genomic area, and we know that there's a limitation when it comes to performing experiments to confirm this theory. However, this is a hypothesis we'll be moving on with. So for our methodology, it's mainly based on a paper that synthesizes by Thompson. I think synthesizes how to recognize the same species only based on genome comparisons. For this, we use several indexes, right? We use the ANI, we use marker genes like the 16S subunit, we use the amino acid identity, and we also can use the DNA hybridization index. So mainly a lot of comparisons to check out what's the deal between the comparison of the Bear 097 genomes and the other Bartonella uniformis genomes. We also added a functional comparison, since it doesn't really belong in the in a taxonomic discussion, let's say. However, we try to keep finding differences between these groups inside the Bartonella bacilliformis species. So what we've got is 47 new sequenced genomes of Bartonella bacilliformis, from which these are all the Bernoulli 97 genomes. So now, after this work is published, we will the public repositories will have more Bernoulli 97 genomes to study from. So probably this will also help to keep checking what are the differences in the first lineage. We can also see we've obtained a Bartonella cachensis genome. This is another species that belongs to the first lineage. It's also very it's not that discussed here in our work and in general in the literature. However, it's still a very interesting species to look for. And finally, we do have more Bartonella bacilliformis genomes incorporated to the overall repository of Bartonella bacilliformis in the NCBI. So, when it comes to the clustering using ANI, we can show that there are like two clusterizations inside the Bartonella bacilliformis species. So, for ANI, the threshold is usually between 95 and 96. Anything that goes further than that is considered to be the same species, and below that is considered to be another species completely. What we obtain with the Bernoulli 97 is that when we compare it to other Bartonella bacilliformis, we obtain this color here. This is a color that reflects that it's actually null 94, the identity, or in any case a 94% of identity with Bernoulli 97 compared to the other Bartonella bacilliformis strains. When it comes to comparing them to each other, what we see is that we have two clusterings. It's the clustering of the Bernoulli 97 and the clustering of the other Bartonella bacilliformis strains. We also made a comparison when it comes to 60ness and also the DDDH. So at my left, what I have is a 60ness comparison added with an AAI, that's the amino acid identity, and on my right, what we have is the DNA hybridization index. So, for when it comes to the 60ness, our threshold indicates that, well, they are not different from each other, all these Bartonella bacilliformis strains, including the Bernoulli 97. Let's remember that 60ness only has a resolution that can reach to a genus level. So, the fact that they still remain the same is very it's very deductible, in a sense that we are still in the same genus. So this Bernoulli 97 does not belong in another group outside the genus Bartonella, and that's something that we can suppose. We could also predict. However, the difference comes when we evaluate it from the horizontal dimension, right? So our threshold here is anything further than 95.38%. So anything that goes beyond here is, we are talking about the same species, but anything that goes below that, we are talking about a different species. And that's also what we are seeing here. This is our Bernoulli 97 group, and these are the other Bartonella strains. By the way, I can also see that there are two groups here, and it's something we will discuss further in the presentation. What we can also see here is the index, the DNA index. This one was very special for us because this one could actually predict if we are talking about maybe a subspecies or some taxonomic level like that. And what we got from here is the fact that no, the threshold still that was indicated for supposing that they could be a subspecies was not reached. What we could conclude from this DDH is the fact that um, they are not the same first and foremost. The bear null 97 is not a subspecies of Bartonella siliformis species. This is the pangenome we've built using the Panarule tool, and this is my main focus here. This rectangle here, in which we show the bear null 97 group specifically. And what we can see is that although there's a pattern here, we can see that there's a huge block here that belongs to all the Bartonella siliformis strains. We see that there are some missing genes in the presence-absence matrix. We can also see there's some additional genes here. We have selected these genes. We know which ones they are. They're in a table specifically. But we haven't really detected a pattern here of any enhanced ability. It should also be mentioned that most of these genes have been labeled as hypothetical according to the annotation software we used. So there's something else that should be digged up here, right? This is a genome alignment. It's a very preliminary one since we are still working on the bear null 97 chromosome close-up. But what we can see here, and for me what's the biggest message really, is the fact that the bear null 97 has a very smaller genome compared to the KC 584. We can also see that there are no like blocks signalizing the alignment. It's more like fine fibers, right? With a very minimal speciation between each other. Although this is not the final alignment, we can see that there's something indicating that there might be like several little deletions between these genomes, and that's mainly the comparison we're doing here. We have also decided to do a comparison between Bartonella cashensis and Bartonella siliformis KC584. And well, these are very, very different genomes from each other. That's our main conclusion here. And as I've mentioned before, Bartonella cashensis is a still not well researched strain that we might tackle in the future as long as we get more strains from it, more isolates in general. This is also a comparison between Bartonella cashensis, Bartonella siliformis KC584, and bear null 97. And what we can see here mainly is the fact that we are talking about distinct alignments when it comes to what we've seen in Bartonella cashensis compared to the main Bartonella siliformis strain, and also what we see with the Bartonella siliformis strain compared to the bear null 97. It's all the fine fibers I was mentioning earlier. We have also done a functional comparison, mainly based on virulence genes. Here are the main categories. We have done basically a category comparison, really. So there are three main virulence categories when it comes to Bartonella siliformis: is the adherence, the motility, and the heme-binding protein. These are all categories given by the database Virulence Finder. What we can see here is the fact that we are searching for the fact that the topography might be different, and we might see patterns or distinct patterns, let's say. So for example, in the case of the category adherence for the virulence or the functional analysis, what we see is that the groups maintain themselves, right? So we see that the bear null 97 group is very maintained, very concise, and the same goes to the rest of Bartonella siliformis. But when it comes to, for example, comparing motility, what we find is very different from what we find it previously, because the bear null 97 groups, we can see it separates from each other. These are, for example, some bear null 97 strains that for the motility comparison, they group themselves with another group of Bartonella acidiformis that are very particular. These are the ones that I've mentioned before, that they, although they still remained as Bartonella acidiformis, they weren't really the same. It's not as if this group is a whole new group. From what I could gather from trying to search what's the difference between this particular group and the rest of Bartonella acidiformis strains, is the fact that all of them possess prophage regions, and mostly the same prophage regions. So I suspect this is, and when you do the pangenome and do the gene presence absence comparisons, what we see here is the fact that these prophage regions are the ones that are highlighted in these comparisons. They are very present, and these prophage regions are also very intact according to the fastest tool. So, well, that's happening inside the Bartonella species, the Bartonella acidiformis species group too. Only side of it is a variation inside the group. There's no comparison we've done here that would show that this group is different. It's completely different from Bartonella acidiformis. But as I was mentioned earlier before, diverging to this particular group here is the fact that we see that Bartonella acidiformis Virulence 97 divides itself into groups here, one here and one that's compared to the other Bartonella acidiformis strains, when it's regarding motility. When it comes to heme binding proteins, as I say, the heme group that's very important to the cycle of Bartonella acidiformis, we also see the same pattern. We see that the Virulence 97 group separates itself and forms a group here and another group here. So the patterns are not similar to the core genome at all. Last but not least, we've been doing several comparisons between several proteins, and what I was trying to find is if there's more convergence here. So there's not necessarily a lot of convergence when it comes to amino acid alignment. However, we can find things like this, like a section that's completely added and different from the sequences completely all along, right? What's curious here, for example, is the fact that we appreciate again the group I've mentioned before, that's kind of tricky and new and not that similar to the other Bartonella acidiformis strains. Here, for example, in the HBV protein, we find that the Virulence 97 group is very particular in the position in which that whole section is deleted, while the other Bartonella strains, they do possess this section here. So we are still trying to do comparisons and trying to see how divergent this can be. So our conclusions is that mainly using comparisons of sequence comparison, we can see that there's a separation between Virulence 97 from the rest of Bartonella acidiformis. We can also see there are distinct patterns existing between the Virulence 97 group and the rest of Bartonella acidiformis when we reconstruct phylogenies associated with the Virulence category. So more studies should be done if we really want to confirm that these are two completely different species, but this is the first study that's trying to address specifically this problem when it comes to Bartonella acidiformis studies. Thanks for your attention.